So this is the title: A genetically guided mechanical wave propagates to drive the formation of an epithelial furrow. So we're in the lab, we're particularly interested in epithelial furrowing. And epithelial furrowing, it's a shape transformation that actually allows cells to move into the third dimension. And this is important because if cells here shown in pink, if they have to form inner organs, well, they need to be then displaced to the inside so they will form the right organ at the right position. And that process is actually called gastrulation during embryo development. Now, if you have a fold, which is forming along a line, this fold is then named a furrow. And actually, we have a lot of furrowing during embryo development. And here I will give you some examples. For instance, we have furrowing during neurulation. And what I'm showing you here is actually the Xenopus laevis, which is the frog embryo. And what you see is this is the dorsal side, which is forming the neural tube. We have also a furrowing during embryo gastrulation. This is an example taken from the Drosophila melanogaster. And again, from Drosophila is actually the shaping of the body. So this is the first future leg of the Drosophila. And this, you will see that furrowing allows to create segments along the leg. What is also furrowing is also interesting because misfurrowing also leads to lethal or severe diseases like spina bifida. This is quite known, but also more severe like craniovertices on an encephaly. So when we look live at furrow formation in vivo live, what you can see is that often furrow formation happens via a fold propagation process. So you can see here during chick neurulation, furrow forms in this mid zone and eventually starts to propagate posteriorly. So in the lab, we therefore asked what are the mechanisms driving fold formation, and eventually also the propagation of this fold in order to drive eventually a furrow. So this project has been developed by Anna, which is a very talented postdoc in the lab. So Anna, she actually decided to study the cephalic furrow, which is a furrow in the developing Drosophila melanogaster, in the fruit fly embryo, that travels along the dorsal ventral axis of the embryo and separates the head tissue of the embryo from the trunk tissue. So here I'm showing anterior on the left side, posterior on the right side, dorsal and ventral, top and bottom. So this cephalic furrow is actually under the control of specific genes that have been already studied and unraveled in 1997. And what is interesting to know is that the cephalic furrow actually forms in cells that are expressing a gene called buttonhead and Eve. So it's where these two genes are overlapping is where actually the cephalic furrow is also forming. If we now look at this process along coronal sections, which are the sections that divide the ventral part from the dorsal part, this is actually what you see. So this is the furrow that it forms. And interestingly, what we can see is that there's actually one cell called initiator cell that what it does is starts to shorten to initiate the formation of the cephalic furrow. So there's a phase of initiator cell shortening, and a second phase called of rolling over, where also the neighboring cells eventually will roll over inside of the tissue. So what you can see here is a 3D segmentation of this process, where I'm showing you here in pink the initiator cell, which is initiating the process of cephalic furrow formation. So Anna, she has been quantifying, in fact, the movement of these cells, and more specifically, she has been tracking the vertices of all these cells, starting from the initiator cells towards the head side or towards the trunk side. And interestingly, what she could see is that the cells, the vertices that moved the faster were the closest to the initiator cell, as you can see here. And this supports already an idea that probably the forces which are generating the changes in shape are generated within the cephalic furrow region, and they're not generated outside of the cephalic furrow region. So in order to test the possibility that tectonic buckling is driving cephalic furrow, which means actually head tissue and trunk tissue, which is actually pushing on the cephalic furrow region to drive the fold, Anna, she devised a technique which is based laser cauterization, thanks to which we can actually stitch and block the cells in order to generate fixed boundary. And by doing so, we're expecting, therefore, if we now isolate the zone where the cephalic furrow will form, if this is driven by tectonic buckling, then in this case, we could block cephalic furrow formation. Nevertheless, when we see at the time lapse, what we can see is that the cephalic furrow can still form, just like in the wild type. And in addition, by forming, it can even rip the tissue away from the zone where the cells were stitched. And this really shows that the forces which are generating the cephalic furrow are generated within the furrowing region. Okay, this is the movie. So Anna then she started to actually explore different ROGAF factor that could eventually drive the formation of the cephalic